Chapter 10 in your textbook covers precipitation and agglutination reactions. In this chapter, we'll cover antigen-antibody binding, precipitation curves, light scattering techniques, passive immunodiffusion techniques, electrophoretic techniques, agglutination reactions, instrumentation for these methods, and quality control and quality assurance. Antigen-antibody binding plays an important role in laboratory in diagnosing various diseases. Immunoassays have been developed to either detect antigen or antibody, and they vary from easy manual tests to more complex automated assays. The first of these assays were based on principles of precipitation or agglutination. With these methodologies, the primary union of binding sites on an antibody with specific epitopes on an antigen depends on two characteristics of antibody known as affinity and avidity. Affinity is the initial force of attraction that exists between a single Fab site on an antibody molecule and a single epitope or determinant site on a corresponding antigen. The strength of attraction depends on the specificity of an antibody for a particular antigen. One antibody molecule may initially attract different antigens, but it is the epitope shape and the way it fits together with the binding sites on an antibody molecule that determines whether the bonding will be stable. Antibodies are capable of reacting with antigens resembling the original antigen that induce antibody production, which is known as cross-reactivity. The more the cross-reacting antigen resembles the original antigen, the stronger the bond will be between the antigen and the binding site. When the affinity is higher, the assay reaction is more sensitive because more antigen-antibody complexes will be formed and visualized more easily. In Figure 10.1 in your textbook, you can see how affinity is determined by the three-dimensional fit and molecular interactions between antigenic determinants and one antibody's binding site. The antigenic determinant on the left has a better fit and charge distribution, which has higher affinity, in contrast to this antibody on the right, of which the antigenic site does not fit as well, and the charge distribution does not allow for good binding. Avidity represents the overall strength of antigen-antibody binding and is the sum of the affinities of all the individual antibody-antigen combining sites. Avidity refers to the strength with which a multivalent antibody binds a multivalent antigen and is a measure of the overall stability of an antigen-antibody complex. Avidity, in other words, is once the binding has occurred, it's the force that keeps the molecules together. High avidity can actually compensate for a low affinity. All antigen-antibody binding is reversible, and it's governed by the law of mass action. This law states that free reactants are in equilibrium with bound reactants, and the equilibrium constant K represents the difference in the rates of the forward and reverse reactions according to this equation. The equilibrium constant K is equal to the concentration of the antigen-antibody complex divided by the concentration of free antibody times the concentration of free antigen. The value of K depends on the strength of binding between antibody and antigen, and the higher the value of K, the larger the amount of antigen-antibody complex, and the more easily visible or detectable the reaction is. In addition to the affinity and avidity of the antibody involved, precipitation depends on the relative proportions of antigen and antibody present. Optimum precipitation occurs in the zone of equivalence. Precipitation reactions involve combining soluble antigen with soluble antibody to produce insoluble complexes that are visible. In order for precipitation reactions to occur, they require that the antigen and antibody have multiple binding sites for one another, and they're equal to the relative concentration of each. As mentioned before, in order for the complexes to be detectable, precipitation reactions have to run into the zone of equivalence, in which the number of multivalent sites of antigen and antibody are approximately equal. In this zone, precipitation is the result of random reversible reactions, whereby each antibody binds to more than one antigen and vice versa, and it forms a stable network or lattice. As can be seen on the precipitation curve on the previous slide, precipitation declines on either side of the equivalence zone because of an excess of either antigen or antibody. In the case of antibody excess, the prozone phenomenon occurs, and this is where antigen combines with only one or two antibody molecules and no cross-linkages are formed. In the prozone, usually only one site of an antibody molecule is used, and many free antibody molecules remain in the solution. In terms of laboratory testing, a false negative reaction may take place in the prozone because of the high antibody concentration. If it's suspected that the reaction is a false negative, diluting out excess antibody and performing the test again may produce positive results. At the other side of the zone, where there is an excess of antigen, post-zone phenomenon occurs, in which small aggregates are surrounded by excess antigen. Again, in this scenario, no lattice network is formed, but in this case, every available antibody site is bound to a single antigen, and no cross-links are formed. For precipitation reactions to be detectable, they need to be carried out in the zone of equivalence. In the post zone with excess antigen, this may obscure the presence of a small amount of antibody. Typically, a test would be repeated with an additional patient specimen taken a week later. The extra time would allow for further production of antibody in the patient, and if upon repeat the test is negative, then it's unlikely that the patient has that particular antibody. Table 10.1 in your textbook provides a concise summary of the different precipitation techniques that we'll be discussing on the subsequent slides. The first two precipitation reactions we'll discuss involve measurement by light scattering. So when antigen and antibody solutions are mixed, the antigen crosslinks with numerous antibody molecules and the lattice networks become so large that they precipitate out of solution. These precipitates in fluid can be measured by means of turbidimetry or nephelometry. Turbidimetry is the measure of the turbidity or cloudiness of a solution. With this method, a detection device is placed in direct line with an incident light, collecting the light after it has passed through the solution. The device then measures the reduction in light intensity caused by reflection, absorption, or scatter. With turbidimetry, the measurements are recorded in absorbance units and are made using a spectrophotometer or an automated clinical chemistry analyzer. Nephelometry measures the light that is scattered at a particular angle from the incident beam as it passes through a suspension. The amount of light scattered is an index of the solution's concentration. If a solution has excess antibody, adding increasing amounts of an antigen results in an increase in the antigen-antibody complex and then an increase in light scattering. The relationship between antigen concentration, as indicated by the complex formation and amount of light scatter, will form a straight line if plotted on a graph. Although the sensitivity of turbidity methods have increased over time, nephelometry is still considered the more sensitive method with a lower limit of detection. Nephelometry also provides actual concentrations instead of absorbance readings, as turbidimetry does. The precipitation of antigen-antibody complexes can also be determined in a support medium such as gel. Agarose, a purified high molecular weight complex polysaccharide that's derived from seaweed, is used for this purpose. So a couple other techniques we'll look at are passive immunodiffusion techniques. When antigen and antibody diffuse towards one another in a gel matrix, visible lines of precipitation will form. When no electrical current is used to speed up the process of antigen-antibody combinations diffusing through gel, this is known as passive immunodiffusion. Radial immunodiffusion is a single diffusion technique that's been used in the clinical laboratory. In this method, antibody is uniformly distributed in the support gel and antigen is applied to a well cut into the gel. As the antigen diffuses out from the well, antigen-antibody combination occurs, and this does in changing proportions until the zone of equivalence is reached and a stable lattice network is formed in the gel. The area of the ring obtained is a measure of antigen concentration that can be compared with a standard curve obtained by using antigens of known concentrations. This figure shows an example of radiodiffusion. So with this technique, developed by Mancini, it's also known as endpoint method. Antigen is allowed to diffuse to completion. When equivalence is reached, there's no further change in the ring diameter. Equivalence occurs between 24 and 72 hours, and the square of the diameter is then directly proportional to the concentration of the antigen. A graph is obtained by plotting the concentrations of standards on the x-axis versus the diameter squared on the y-axis, creating a smooth curve to fit the points. So here we have the standards 1, 2, and 3 on the glass slide, along with the unknown patient sample. And here you can see that the standards have been plotted on the graph, and then the patient unknown concentration can be determined based off the standard curve or plot. One of the older classic immunochemical techniques is the Agglutination double diffusion. So with this technique, both antigen and antibody diffuse independently through a semi-solid medium in 2 dimensions, horizontally and vertically. Wells are cut into the gel, and reactants are added to the wells. Most Agglutination plates are set up with a central well surrounded by 4 to 6 outer wells that are equal in distance. Antibody that is multi-specific is placed in the central well, and then different antigens are placed in the surrounding wells to determine if the antigens share identical epitopes. Diffusion takes place radially from the wells, and after an incubation period between 12 and 48 hours, the antigen meets the antibody, and the point where the zone of equivalence is reached can be determined. The density of the lines reflects the amount of the immune complex formed. The 3 possible patterns that can be observed on the Agglutination diffusion technique are identity, partial identity, or non-identity. In this diagram, there's 3 examples of the patterns that could be observed. In figure A at the top, if the antigens are identical, they will react with the same antibody and the precipitate line forms a continuous arc. And this is identified as serological identity. In figure B on the left, the antigens share no identical determinants, so they will react with different antibodies and 2 cross lines are formed. So an X can be observed, and this is identified as non-identity. In figure C on the right, if antigen has 3 determinants in common with antigen 1, one of the antibodies reacts with both antigens. another antibody that reacts with different determinants on antigen 1 passes through one precipitation line and forms the spur on the yellow line. The spur formed always points to the simpler antigen with fewer antigenic determinants, and this is identified as partial identity. Diffusion techniques can be combined with electrophoresis to speed up or sharpen results. Electrophoresis separates molecules according to differences in their electrical charge when they're placed in an electric field. A direct current is forced through the gel, causing antigen, antibody, or both to migrate. As diffusion takes place, distinct precipitin bands are formed. Immunoelectrophoresis is a double diffusion technique that incorporates electrophoresis to enhance results. As interpretation is difficult with immunoelectrophoresis, immunofixation electrophoresis has largely replaced the initial method, and this gives faster results and is easier to interpret. With immunofixation electrophoresis, it has similar properties as immunoelectrophoresis, except that after electrophoresis takes place, antiserum is applied directly to the gel surface rather than placed in a trough. Agarose or cellulose acetate can be used for this purpose. Immunodiffusion takes place in a shorter time and results in higher resolution than when antibody diffuses from a trough. Most often with immunofixation electrophoresis, an antibody of known specificity is used to determine whether patient antigen is present. The unknown antigen is placed on gel, electrophoretic separation takes place, and then reagent antibodies applied. Immunoprecipitates form only where there's specific antigen-antibody combinations that have taken place, and the complexes are trapped in the gel. Typically, patient serum is applied to 6 lanes of gel. After electrophoresis, 5 lanes are overlaid with one of the following antibodies: antibody to gamma, alpha, or mu heavy chains, and to kappa or lambda light chains. The 6th lane is overlaid with antibody to all serum proteins, and this serves as a reference lane. This method is particularly useful with serum, as it's helpful in determining over or underproduction of particular antibody types. This figure 10.7 in your textbook shows an example of an immunofixation electrophoresis gel. So here, a complex antigen mixture, such as serum proteins, is separated by electrophoresis, and then an antiserum template is aligned over the gel. Protein fixative and monospecific antisera, IgG, IgA, IgM, kappa, and lambda, are applied to the gel, and after incubating for 30 minutes, the gel is stained and examined for the presence of paraproteins. Precipitates form where specific antigen-antibody combination has taken place. So in this example, we have our reference lane on the left, and in this case, the patient has an IgG monoclonal antibody with lambda chains. So here you see there's a line of precipitate under the G row and under the lambda row, indicating, as mentioned, the patient has IgG monoclonal antibody with lambda chains. So up to this point, we've been discussing the principles of precipitation reactions. So where these reactions involve soluble antigens, agglutination is the visible aggregation of particles caused by a combination with specific antibodies. Antibodies that produce such reactions are often called agglutinins. And because this reaction takes place on the surface of the particle, antigen must be exposed and able to bind with antibody. Types of particles that participate in such reactions include erythrocytes, bacterial cells, and some inert carriers such as latex particles. Agglutination, like precipitation, is a two-step process that results in the formation of a stable lattice network. The first reaction is called sensitization, and this is the initial binding that involves antigen-antibody combination through single antigenic determinants on the particular the particle and is rapid and reversible. The second step, or lattice formation, is the formation of cross-links that form the visible aggregates. And this represents the stabilization of the antigen-antibody complexes with the binding together of the multiple antigenic determinants. This figure demonstrates the phases of agglutination. So initially you have your antibodies separate and your antigens with multiple determinants. Once these are combined, sensitization occurs in which there's no physical reaction. But although there's no visible reaction, the antibody-antigen combinations start to join by single antigenic determinants. The second phase, lattice formation, is the visible agglutination phase, where these large aggregates form that have this visible agglutination. Agglutination reactions are fairly easy to carry out. They don't necessarily require complicated equipment, and they can be performed as needed in the laboratory without having to batch specimens. There's various kits available for standard testing, so reagent preparation is minimal, and agglutination reactions are frequently used as serological testing, in which they can identify either antigen or antibody. Typically, most agglutination tests are qualitative, in which they indicate only the absence or presence of antigen or antibody. But there are scenarios where dilutions can be made to obtain semi-quantitative results. Of the types of agglutination reaction, there's direct agglutination, and this occurs when antigens are found naturally on a particle. So one of the best examples of direct agglutination testing involves known bacterial antigens used to test for the presence of unknown antibodies in a patient. One example is the Weidel test, which is a rapid screening test used to help determine typhoid fever. So a significant finding with this test is a 4-fold increase in antibody titer over time when paired dilutions of serum samples are tested with any of the antigens. There are now more specific tests, but the Weidel test is still considered useful in diagnosing typhoid fever in developing countries and does remain in use in many of the areas throughout the world. If an agglutination reaction involves red blood cells, it's called a hemagglutination reaction, and the best example of this occurs with ABO blood group typing of human red blood cells, and this is one of the most frequently used immunoassays. In this testing, patient red blood cells are mixed with an antiseria of the IgM type and can be used to determine the presence or absence of the A and B antigens. This reaction is usually performed at room temperature without the need for enhancement techniques. Figures A and B on the slide depict hemagglutination reactions and how grading of the agglutination reactions is done. So in figure A, it's a tube method. So if the tubes are centrifuged and shaken to suspend the button, reactions can be graded from negative to 4+, depending on the size of the clumps observed in the tube. Alternately, a rapid slide method could be used, in which a negative has an even suspension with a cloudy background, to a strong agglutination reaction in which large clumps and a clear background are observed. Another type of agglutination reaction is passive agglutination. Passive, or also known as indirect agglutination, employs particles that are coated with antigens not normally found on their surfaces. A variety of particles are used, including erythrocytes, latex, and gelatin. The use of synthetic beads or particles provides the advantage of consistency and uniformity. These reactions are easy to read visually and give quick results. Passive agglutination tests using latex agglutination have been developed for antibodies to group A strep and to viruses such as rotaviruses, cytomegalovirus, rubella, varicella zoster. Because many of these kits are designed to detect IgM antibody, and there's always the risk of non-specific agglutination caused by the presence of other IgM, these reactions need to be carefully interpreted. So with these commercial test kits, they're usually performed on disposable plastic or cardboard cards, or sometimes glass slides, and the kits contain positive and negative controls. So if you don't get expected results, the test is then not valid with the controls. These tests are usually screening tests, and then they need to be followed up with more extensive testing or a diagnostic confirmatory test. A few other latex agglutination test examples include those to detect rheumatoid factor or even antibodies to hepatitis viruses and HIV. Again, with this type of agglutination reaction, antigen is attached to the carrier particles, such as the latex particles, and agglutination occurs if the patient antibody is present. Reverse passive agglutination is another type of reaction, and this is commonly used to detect microbial antibodies. These types of reverse agglutination tests are most often used for organisms that are difficult to grow in the lab, or in scenarios when rapid identification will allow treatment to be initiated more quickly. Common applications include rapid ID of infectious agents such as group B strep, Staph aureus, strep group A and B, rotavirus, and Cryptococcus neoformans. And these applications can be used in detecting soluble antigens in urine, spinal fluid, or serum. With the reverse passive agglutination reaction, antibody is attached to the carrier particle, and agglutination occurs if antigen is present in patient samples. This figure just gives a quick summary comparing the types of agglutination reactions, particularly the passive and reverse passive agglutination. So on the top is an example of passive agglutination, in which antigen is attached to the carrier particles, and agglutination occurs if antibody is present in the patient sample, opposed to the reverse passive agglutination on the bottom, in which carrier particles are attached to antibody, and agglutination occurs if antigen is present in the patient sample. The last types of agglutination reactions we'll discuss are agglutination inhibition reactions, and these are based on competition between particulate and soluble antigens for limited antibody combining sites. With these types of tests, the lack of agglutination is an indicator of a positive reaction. Typically, this reaction involves haptens that are complex to proteins. The haptene-protein conjugate is then attached to a carrier particle. The patient sample is first reacted with a limited amount of reagent antibody that's specific for the hapten being tested. Indicator particles that contain the same hapten that one wishes to measure in the patient are then added. If the patient sample has no free hapten, the reagent antibody is able to combine with the carrier particles and produce a visible agglutination. In this case, agglutination is a negative reaction, indicating that the patient did not have sufficient hapten to inhibit the secondary reaction. Hemagglutination inhibition reactions use the same principle, except red blood cells are the indicator particles. So this type of test has been used to detect antibodies to certain viruses, such as rubella, influenza, and RSV. So the red blood cells have naturally occurring viral receptors. When the virus is present, spontaneous agglutination occurs because the virus particles link the red blood cells together. Presence of patient antibody inhibits the agglutination of that reaction. These diagrams depict a positive and a negative inhibition reaction. So in the top example, reagent is added to the patient sample. If patient antigen is present, the antigen-antibody combination results. So then when the antigen-coated latex particles are added, no agglutination occurs, which indicates a positive test. If no patient antigen is present in the sample, the reagent antibody combines with latex particles and agglutination results, and this indicates a negative test. With the hemagglutination reactions, in the presence of certain viruses, red blood cells will spontaneously agglutinate. So if patient antibody is present, as in the samples at the top, agglutination is inhibited. So there would be no agglutination, which indicates a positive test for the antibody. In a patient sample that lacks antibody, when the antigen is added, agglutination reacts with the red blood cells, and this is a negative test. This table, although not available in your textbook, is a concise reference for comparing the agglutination reactions. So it outlines the type of reaction, whether it's direct, indirect, reverse, passive inhibition, or hemagglutination inhibition, the principle of each method, and the expected results. So use this as a tool, especially as you're studying. Although agglutination reactions don't require complicated instrumentation, several chemistry analyzers have been developed using automation to increase sensitivity. Nephelometry has been used to the reading of agglutination reactions, and the term particle-enhanced immunoassay is used to describe such reactions. One type of instrumentation system is called particle counting immunoassay. This method involves measuring the number of residual non-agglutinating particles in a specimen. These particles are counted by a laser beam in an optical particle counter, similar to methods used to count blood cells. They measure the forward light scatter, and very large and very small particles are excluded by setting certain thresholds on the instrumentation. This type of method has been used to measure serum proteins, therapeutic drugs, tumor markers, and certain viral antigens. The last topic discussed in this chapter involves quality control and assurance, particularly for these precipitation and agglutination reactions. Because even though these are fairly simple to perform, interpretation has to be done carefully. Techniques need to be standardized regarding the concentration of antigen, incubation time, temperature, diluent, and the method of reading, and taking note of possible cross-reactivity and interfering antibody. Most cross-reactivity can be avoided by using monoclonal antibody directed against an antigenic determinant that's unique to a particular antigen. Other considerations include proper storage of reagents and attention to expiration dates. Each new lot should be evaluated before use, and manufacturer instructions for each kit should always be followed. The sensitivity and specificity of different kits may also vary, and this needs to be taken into account. Major advantages of agglutination reactions include how rapid they are and how they are fairly sensitive, and that samples containing an organism does not need to be viable in order to test positive. Although there are advantages to testing, it's important to be aware that a negative result does not rule out the presence of the disease or antigen. Major points discussed in this chapter include precipitation involving the combination of soluble antigen with soluble antibody to produce insoluble complexes that are visible, and these again are the basis of many laboratory tests. The combination of antigens and antibody depends on the affinity between one antibody binding site and a single epitope on an antigen. Avidity is the sum of all attractive forces occurring between multiple binding sites on antigen and antibody. The zone of equivalence is when concentrations of antigen and antibody yield maximum binding. So this is where the multivalent antigen and antibody sites are approximately equal. Prozone indicates antibody is in excess and precipitation or agglutination reactions won't be observed. Postzone is when antigen is in excess, and similarly, precipitation or agglutination would not be observed. Precipitation techniques include nephelometry, passive immunodiffusion techniques, including radial immunodiffusion on Octoloni double diffusion, and electrophoretic techniques. The process of agglutination is divided into two steps, sensitization and lattice formation. In direct agglutination, antigens are found naturally on the indicator particle. In passive agglutination, antigens are artificially attached to a particle. Reverse passive agglutination is called this because antibody is attached to the indicator particle. And finally, agglutination inhibition is based on the competition between the antigen code particle and soluble patient antigen for a limited number of antibody sites. So this is the only scenario of the different testing methods we talked about in which agglutination represents a negative test.